By way of disclosure, as Crystal noted, I am a co-founder at Juno and have several relationships with Juno that are appropriate to disclose. So what I'd like to do is to tell you about our program here in Seattle that we really kicked off about 4.5 years ago in earnest at the Ben Towne Center, where we try to encapsulate kind of the full spectrum of the translational research process with discovery labs. We have clinical production process development teams, we have a GMP production, and then we have a correlative biology group that analyzes specimens from treated patients. So it's a great time to be in immunotherapy. I remember the immunotherapy meetings, oh gosh, 10 years ago, where there'd be 4 of us, we would be presenters in the AV team, and we'd kind of be encouraging us to go forward. But these are new times, and really the excitement has revolved around both the ability to inhibit checkpoints, to unleash immune responses that are dormant in patients. And then the second area that my lab has focused on for many years has been on the approach of adoptive T-cell therapy. And unlike approaches that have previously been employed that utilize tumor-resident TILs that are basically re-isolated ex vivo and adoptively transferred after lymphodepletion, and this was really a major contribution of the surgical branch at the NCI to provide us that insight, my lab has been interested in the development of synthetic antigen receptors for applications in the clinic. And so I would say, over the last 20+ years, how did we get to this inflection point? I wish I could say there was a clear linear effort that we knew what we were doing in the beginning and we just had to march through, but there was a lot of empiricism and trial and error, and maybe some good fortune for the field a couple years back when we first started seeing responses in leukemia patients. But a lot of our system is still quite empiric. Many chimeric antigen receptor designs are put together, and there's many elements of a CAR that influence its signaling. Our vector transduction can lead to variable numbers of cells that are active in various different levels of transgene expression that can influence the behavior. And then for most of the field, we've been transducing bulk populations of T cells that are in PBMC preps, in which some of the T cells may have no net effect, some positive effect, but some could have negative effects in terms of toxicity. So our current state of affairs, I think, with CAR T-cell therapy utilizing an idealized leukemia patients as follows. A very small dose of T-cells is administered to a patient, usually with demonstrable disease that is expanding. These T-cells need to proliferate in multi-log fashion, and they reach a tipping point where there's enough T-cells and activity that you start to see this very abrupt fall off in leukemic cell burden and normal B cell burden, and you get a B cell aplasia. And ideally, this response has a duration that is necessary for sterilizing immunity or eradication of resident leukemic cells throughout the body. And there is a waning of this persistence, and at some point, you have reemergence of normal B cells because your CAR T cell activity has waned to that point. And that's probably important for most children getting CAR T cells for ALL, that they do reconstitute a B cell repertoire. So that's the ideal state, but a number of things still don't go perfectly in the clinic in a reliable way, and I think we need to move this therapy into more of a drug-like effect in terms of what we can expect in terms of efficacy and safety. So the first thing that can go wrong is, for a given patient, there's a product that has a composition that is unable to do that multi-log expansion. At the peak of these responses, we are seeing tremendous, in some patients, very serious toxicities, and this is going to be a challenge to the field to get this therapy out into more community-based settings. We're already seeing that under the pressure of a unifocal CAR T-cell response, that tumor cells will emerge in terms of escape variants that could lead to relapse. And then finally, we have this issue of that we currently can't control the duration of our CAR T-cells, and there may be patients that need a controlled ablation. So I think my lab is trying to embrace an approach that utilizes the tools of synthetic biology and the kind of the way in which engineers and synthetic biologists approach development of new or orthogonal biological outputs. And we're doing this in the context of T cells. And so it's really, you know, understanding, and I'm still in the learning phase, you know, we're building parts, we're putting together devices, putting them into modules, and then modeling them, testing them, and seeing how it agrees with the model. And this is very intuitive for how the electronics industry has evolved over time, but it's still a little bit new for mammalian cell engineering. So synthetic biologists use some terms that I might refer to. Parts are kind of the widgets, the transistors, the capacitors. For us, it may be an open reading frame and a protein, a promoter, a rheostat. Parts are assembled into devices. This could be, you know, a regulated promoter with a transgene. And then those devices are engineered into a chassis, and the chassis for us is T cells. So starting with this first issue regarding reliable engraftment, what we've done is take the approach that we will fractionate the PBMC prep coming from an apheresis for a patient into defined subpopulations of cells, and we will selectively engineer them and then be able to infuse those subpopulations in graded doses or ratios. So, you know, our PLAT2 trial at Seattle Children's, we are using a defined population of 4s and 8s at a 1-to-1 ratio. These are some of the attributes of the vector system in the CAR. And then, as Olivia yesterday alluded to, we've engineered in this cell surface barcode for various applications that I will share with you. So this means that we can deliver doses of cells that have uniform purity for their phenotype or their cell source. The first basic is how many 4s and how many 8s. We infuse them at a 1-to-1 ratio, and they've been purified, so 100% of these cells express a fairly tight, calibrated level of transgene. We do this by doing an upfront purification on the apheresis product. We positively select the CD4s and CD8s. After that, it can go into manufacturing, or we can cryopreserve an intermediate. So if the manufacturing production is overloaded, we can wait a couple of days until a manufacturing slot opens. These cells are this is fairly standard for the field, that we bead stim them, we transduce them 2 days later, but what we do in the mid-process is we come back in with the Clinimax and we purify the cells on our EGFR T tag. And for most products, they're made in about 12 days. So what this means is our prior protocol, where we didn't define cell composition, you could see some products might have trivial amounts of CD4s, some patients may have more, so it's really quite variable from product to product, from patient to patient, and you can see that now it becomes homogeneous for phenotype of the cells. Likewise, transduction levels and how many cells express the transgene and what levels becomes normalized too, so it becomes a fairly reproducible product. And this product has some characteristics based on the cytokine cocktails that we grow the 4s versus the 8s in that have marker sets like CD62L and CD28 and CD28 expression. This is at the end of culture, not the beginning of culture. So these cells have a number of markers that are associated with high replicative potential. And when we modeled these different product types in our Raji model system, NSG mouse, you could see that if we use an undefined product in terms of how many 4s and 8s and what percentage of CARs, we get this type of anti-tumor effect. If we just isolate on 4s and 8s and not CAR, we get an intermediate effect. And likewise, if we purify in CAR, but the 4s and 8s are not defined, we get this. And it's the efficacy of the defined product for both 4 and 8s and CAR expression that in this model system gave us the highest potency. So Rebecca Gardner in Julie Park's phase 1 program at Seattle Children's, we launched a phase 1/2 trial, PLAT2. And to date, and this is rolling data, but to date we've enrolled 37 patients, 32 are valuable for response. You could see that this complicated manufacturing process, we've had 100% success rate in making the CD8 products, and we had one CD4 product that we couldn't make for a patient. And all of this is without screening patients for a T-cell proliferation assay as an eligibility test to go on trial. So this is, we enroll patients independent of that. So it's a fairly robust manufacturing system. We've looked at cell doses between 500,000 10 million cells per kilogram. And this is the patient profile of patients on our study. Most patients with multiple relapse refractory disease, a high number of post-allo transplant patients, most coming in and getting dosed at a time when they have significant amounts of antigen burden. And I think importantly for our trial, we started our first cohorts of patients getting cyclophosphamide alone lymphodepletion. And I think, you know, finally us folks in Seattle have learned from you folks on the East Coast that have taught us over and over again that fludarabine is important. And we are certainly now evaluating the MTD of the T cells in the context of flu-cy. So for a typical patient, this is 14 days after the administration of 250,000 cells per kilo of CD4 product and 250,000 cells per kilo of CD8 product. And for most patients, they achieve B-cell aplasia, very high numbers of CAR T cells because they express the EGFR-T. We can follow them by flow. We typically and reliably see an asymmetric expansion of the 8s over the 4s. And by day 14, a lot of the T cells have gone through a fairly naive differentiation state at the time of transfer. They, day 7, become effector cells driven by antigen, but by day 14, the CAR T cells, most of them have achieved a memory cell state with high numbers of central memory cells in the CD8s, and the CD4 cells have a higher proportion of effector memory cells. And we see these cells in the CSF at the same proportion that they are in the blood. So for an audited data set, and this is an intent to treat, so if you sign an informed consent and are eligible, we count you. And this is our waterfall plot, if you will, for our MRD negative CR rate. So we have now a 91% at 32 patients, and these are audited data on the first 22. High disease burden doesn't seem to be a barrier for achieving a CR state. We've had I'm sorry, this is the high burden, and that was the dose. And we've had a couple patients who are infants at the time they were diagnosed and have been through therapy, and we have been able to, in both of those patients, it's limited, but we've been able to make product, and those patients, those infants, have had CRs. These are the kind of the pharmacokinetics of our cells, and as Olivia pointed out yesterday, it doesn't fall into discrete categories based on T-cell dose and disease burden, as well as it does if you calculate what I'll call the CD19 antigen burden of the patient, which is it's the sum of the normal B cells and the leukemic T cells. And these are fairly similar whether it's the CD4 cells or the CD8 cells that engraft. So here's the relationship between induction of an MRD remission, the sustained duration of B-cell aplasia, and the durability of that remission. And certainly we are encouraged by patients like this that are out, you know, over 200 days with both B-cell aplasia and remission. But we are seeing some patients that have short durations of B-cell aplasia that go on to relapse, typically with CD19 expressing disease. We've had some relapses where they are still under B-cell aplasia and they relapse with an escape variant. So this is kind of our Kaplan-Meier of B-cell aplasia. And Steve, I think you asked Olivia, you know, how much time of B-cell aplasia are we thinking is necessary? And this is really, okay, I'm going to go out on a limb, but I would be much more comfortable if this 6-month B-cell aplasia level were up here. So I don't know if it's 3 months of B-cell aplasia or 6 months, but that's kind of our thinking in terms of how long we want to have that bioactive cell product operational in the patient. But for some patients that have short durations of persistence, it's not going to be amenable to redosing or other manipulations because these patients acquire an anti-transgene immune response mediated by T cells. Sandra Dells is mapping epitopes, but most of them are in the murine SCFE portion of the CAR, but not all of them. There's some fusion sites that for certain HLA alleles generate T cell responses. So we have to figure out how to be able to get more durable persistence in patients by intensifying those aspects of the therapy that prevent rejection based on an immunologic mediated mechanism. This is our leukemia-free survival thus far in the trial and our overall survival. And so what we're looking at to basically go try to get more patients to have 100 days or 200 days of B-cell aplasia. As I said, we're intensifying the lymphodepletion. The addition of fludarabine is not an additive, but a synergistic effect on both the magnitude of engraftment, the duration, and it looks like it's fairly significant in terms of inhibiting a rejection response. So redosing has been effective in our adult trials at the Fred Hutch. There's humanized CARs that are getting close to clinical implementation. And then what we're going to do in the next couple of months is to employ a vaccine. So when these cells engraft and become fairly quiescent memory cells, we can infuse into that patient what we call a TAPC vaccine, which is a product we make from the patient where we transduce the T cells to express a CD19 molecule, and we cryopreserve it. And so we're going to look at repetitive dosing of a vaccine, not only to increase the numbers of the T cells, ideally, and to increase their duration of persistence in the patient, but we also think that these quiescent memory cells go to their memory niches, and they're maybe not the same cell that can get into the CNS or another site where a leukemic cell is laying dormant. So by activating the cells recursively, we think we're redistributing them into tissue beds, and that may be important. So that's coming. So this next part of kind of the idealized system that can get derailed is the toxicity problem. And certainly we have patients, because our current system is constitutively on, the gas pedal's on, it's a constitutive promoter. You put these cells in and you stand back and you hope, the patient gets into orbit safely. But a lot of patients have a struggle for that. We have not had any fatalities yet. And we have some patients, 90+% of their CAR T cells at peak or all T cells are CAR T cells, and they have the cytokine storm that most of you are familiar with. So in terms of our toxicities of these 22 audited patients, 8 of 22 have had either serious cytokine storm or serious neurotoxicity, and 2 patients have had the combination of the both. And you see in terms of disease burden, we could have some patients that are MRD negative that get a serious cytokine storm, and they tend to be patients that haven't been heavily pretreated coming into the trial, so they have a lot of B cells. So again, this B cell index or antigen load may be an important variable, and we're still working through that. So what can we do about this? Because being reactionary to a patient in the ICU is less desirable than understanding how we can intervene preemptively to prevent the worst side effects. And so here are 2 patients that we were very worried about in terms of their underlying physiologic status coming into the trial, and we preemptively treated them as they just started their CRS, both with early TOSI and then DEX and more TOSI, and TOSI and DEX really on the upswing of their CAR T-cell engraftment. And what we found is that the CAR T-cell engraftment is very robust in these patients. We didn't collapse the CAR T-cell population. They achieved B-cell aplasia for prolonged periods of time and engraftment. And there is data both in our lab and other labs that these T cells that are being actively triggered through the CAR with all that ERK phosphorylation, that T cells can temporarily assume a state of steroid resistance. And perhaps the major role of corticosteroids for treating this is not to ablate or attenuate the activity of the CAR T cells, it's to blunt the responsiveness of the responding tissues to the cytokines that they're getting as a consequence of the activation of the T cells. So what we want to do is start to form orderly protocols for preemptive therapy and really analyze how we can do this effectively without nipping the therapeutic response in the patient. I think to do this carefully and effectively, we have developed a non-human primate model that's really a phenocopy of CAR T-cell therapy in the clinic. And Agna T, I'll just say T because her last name is impossible for me to pronounce. She's a fellow in the lab that's been working with Leslie Keene, my collaborator at the Ben Towne Center, who's an expert in primate manipulations. And so we have used in this system lymphodepletion with cyclophosphamide, then doses of CAR T cells, typically 10 to the 7th to 10 to the 8th per kilo, and then we follow these animals. And the CD19 CAR, FMC63, and the other ones don't cross-react with monkey CD19. So for this, we use a CD20 CAR. And that CD20 CAR is from the LU16 antibody. You could see LU16 as an antibody binds to both human CD20 and on a macaque LCL line, macaque CD20 and CAR T cells triggered by macaque CD20 positive cells elicit activation responses. And so in this animal, as what we have seen in our first pilot study is, day 14, about 30% of all the T cells in the circulation of this animal are EGFR-T positive.